Let's look at those medulla concentration gradients. Now again, the outer region of the kidneys is the cortex, and the deeper inner region is the medulla. And remember, the deeper you go into that medulla, the higher the concentration of solutes. If you didn't have that high concentration of solutes in the medulla, you wouldn't be able to pull that extra 15% out of that descending part of the loop of Henle. So if you want to concentrate urine, in other words, you want to really concentrate the waste inside of the nephron, you got to pull most of that water out of it, because that's primarily what that filtrate is. So that's going to prevent you from losing a large amount of water. Again, you lose a certain amount of it to balance blood pressure and such, but you can't lose too much. If you went from losing, say, on average, a little less than 1%, and you started to lose that additional 15% out of that loop of Henle, you couldn't maintain blood homeostasis. So the kidneys have got to maintain those high solute concentrations in that medulla. You look towards the outer regions, it's about 300 milliosmoles. Way down deep towards the bottom of that loop of Henle and further, it's around 1200. That's a lot of solutes, giving a large pull out of that descending part of the loop of Henle. If you want to maintain these gradients, you got to have that loop of Henle. You got to have that vasa recta, which is the part of that second capillary, the peritubular, that follows the loop. You got to have that constant recycling of urea. You don't want to pull urea out of the loop, but with that huge volume of water that's pulled out, about half of it is. And that's what accounts for a lot of those solutes in that medulla. So if you want to keep all these concentration gradients, starts with the active transport of the sodium. You're going to have along with it co-transport of other materials like potassium and chloride. And those ions are coming out of that thick portion of the ascending limb. Remember, there's descending and ascending, there's thick and thin. You're going to pull that material into the interstitial fluid, keeping those solute numbers high. You're going to have impermeability of the ascending part of the loop. Otherwise, the water would come right back in. You want to get the stuff out, not in it. You got to have that vasa recta picking up that water as it comes out of the loop. So as the vasa recta goes down into the medulla, it picks up more solutes, and that lets it pull that water from the interstitial spaces. You got to have the active transport of ions from the collecting ducts. Remember, many distal parts of the tube lead into the collecting ducts. You got to have that passive movement of urea. Again, you leave roughly half of it inside the nephron. That's more than enough to maintain homeostasis. But as you pull about half of it out into the medulla, keeps those concentration numbers high. Looking at what's in the urine, again, most of it's water. You got waste materials. Urea is the most abundant of them all. It's where we get the name urine from. Any excess material like ions and trace amounts of hormones and other things. These materials are responsible for a large part of that high osmolality of that medulla. You got these descending limbs of the loops, which are permeable to urea. Lets that material move out. Urea diffuses into the interstitial spaces and adds to those large osmolality numbers. You got the ascending limbs and the distal tubules, which are impermeable to the urea. Keeps it from moving back in. You got the collecting ducts, which are permeable. That's the last parts you see actually after the nephron, leading down towards the very tip of the medulla. You got that urea flowing in a cycle. Some of it moves into the nephron, some of it right back out, keeping those concentration gradients high. And remember again, when you look at these hormones, they work on the nephron, they work on the distal tubule and the collecting ducts. And we'll look back at the renin, angiotensin, aldosterone mechanism. Lots of organs and hormones involved here working together. So what happens is renal blood flow is reduced. Say maybe blood pressure is dropping. These juxtaglomerular cells of the kidneys will secrete renin. The renin will carry out the conversion of angiotensin 1 in the liver. Angiotensin 1 will be converted to angiotensin 2 at the lungs. And that angiotensin 2 is a powerful vasoconstrictor, and it'll stimulate the release of aldosterone. So say again, if blood flow to the kidneys is dropping, maybe it's because blood pressure is dropping. If you constrict blood vessels, that'll raise pressure up, and aldosterone will tell the kidneys to hold water. That'll help to raise it also. So we've looked at aldosterone before. It does several things when it gets to the kidneys. It tells the kidneys to reabsorb sodium, which will cause the kidneys to hold on to water. It'll work to raise your blood pressure up right there. At the same time, it tells the kidneys to excrete hydrogen and potassium. If it pumps those inside the nephron, they'll be lost. So somebody has acidosis or hyperkalemia, it's good to release aldosterone because it tells the kidneys to get rid of that excess material. So all that's good for raising up your blood pressure and helping to balance the hydrogen and the potassium in the body. Remember, ADH works very strongly on the kidneys. ADH will tell the kidneys to reabsorb more sodium. You hold more sodium, you'll hold more water. Again, that'll help to raise your blood pressure. And remember, ADH has a second target tissue, the smooth muscle around blood vessels. It tells it to constrict. So as it tells the kidneys to hold water that raises blood pressure, it'll constrict blood vessels, which raises them too. You look at ANH, atrial natriuretic hormone, works opposite of ADH. Tells those nephrons don't pull as much sodium back into the body. As sodium stays in the nephron, water stays with it, water is lost, and that'll lower your blood pressure there. It's working opposite of those two previous hormones. And again, talking about how much glucose can be reabsorbed in the kidneys. Only about 320 milligrams a minute is going to be the max for your average adult. Somebody has far too much sugar in the blood. Remember, it'll be floating around in the plasma, and that's what's put into these nephrons. So if more than that 320 is put into the nephrons, the transporters can't move it all back. They'll be saturated. There'll just be more of it there than what they can pull back into the interstitial spaces. And if they can't pull that sugar back, some of the sugar will be lost. And as a solute, like sugar is lost that shouldn't, water's going to follow it. And that's why diabetics have such a high urine output. You lose that solute that you shouldn't be losing, the water will follow it. It's what's called osmotic diuresis. So remember, the kidneys never get rid of glucose on purpose. That never happens. It's only when they can't pull it all back that it gets lost. That's completely unintentionally. So there's your pictures of the kidneys and such we had again, and the links to the study guide.